Here is a video that I've made that I call Making Sense of the Chapter 10 Activity Table, or a Walk Through the Antimicrobials. In other words, I'm going to take the information that was in the Chapter 10 Activity Table and I'm going to rearrange it and talk to you about why I'm rearranging it, so hopefully it will help you understand the material better. The first thing I'm going to do is I'm going to rearrange the antimicrobials from the table, and part of that is if I rearrange it, then I can understand it in a different way. In the table I have them alphabetically. Here I'm going to put antibacterial antimicrobials, we also call them antibiotics. I'm going to put antifungal next, antiprotozoal drugs next, then antihelminthic or worming agents, I guess you could call them deworming agents, and then the antiviral, those agents which inhibit the growth of bacteria. Now, I have them grouped this way for a reason, and we'll get into that. Now I have one lonely antifungal, a antiprotozoal, and then I have a second antiprotozoal that you'll notice is also under here as an antibiotic, and we'll get into that in a minute too. And then I have one lonely anti-deworming agent, which, by the way, I have found out is not used in humans in the United States anymore. It's still used in animals. And then we have our two antivirals. So, let's start making sense of all of this information. So, the first thing that I did to make sense of it was to group them by how they act. Now, the next thing that I'm going to do is I'm going to group everything that is a eukaryotic cell. So fungi, protozoa, and worms, which are animals, are all eukaryotic cells. Now, antivirals, I'm going to kind of group over here because viruses, at least for these antiviral agents, work in eukaryotic cells, specifically in animal cells. So they're kind of related to the deworming agents, sort of. Now over here, I have everything that affects prokaryotic cells. Now that I have this organized and I have straightened my mind that these are dealing with inhibiting the growth of eukaryotic cells in the human body, and these are for inhibiting viral replication in eukaryotic cells, specifically us, we have eukaryotic cells. Now we're going to talk about how these all act. And in the process, we're going to talk about why we have so many antimicrobials under here, under antibiotics, that inhibit the action, inhibit the growth, kill prokaryotic cells, and why we have so few that are effective for eukaryotic cells. So let's look at this first grouping, penicillin G, ampicillin, and isoniazid. Now all of these inhibit cell wall synthesis. Well, why don't they inhibit fungal cell wall synthesis? Well, it's because these two, in particular, inhibit something that is in bacterial cell walls that are not in anything else. And what is that? Peptidoglycan. Now, isoniazid is not a peptidoglycan cell wall synthesis inhibitor, but it does work against acid-fast bacteria, inhibiting the mycolic acid, which is embedded in the peptidoglycan, and we never find mycolic acid in anything but acid-fast bacteria. So we're still attacking something that is unique to prokaryotic cells. So let's look at these two, erythromycin and tetracycline. They inhibit protein synthesis. Well, eukaryotic cells undergo protein synthesis. Why do they not inhibit fungi, protozoa, and animal cells? In other words, why doesn't it inhibit your protein synthesis? Well, what is unique about prokaryotic protein synthesis versus eukaryotic cell synthesis? Now we're going to have to go back to chapters 3 and chapters 4 for this. And it's because both of these inhibit 70S ribosome function. They leave alone 80S ribosome function. That's why it's unique to prokaryotic cells, specifically bacteria. Now, for these last two, they attack specific metabolic functions. By specific, I'm talking, well, I should say metabolic functions that are specific to prokaryotic cells. We don't want to attack glycolysis because that'll cause nasty side effects to you. You don't want to attack the Krebs cycle or the electron transport chain for the same reason. So, what metabolic functions are these specific for? Nucleic acid synthesis. Na is my abbreviation for nucleic acid synthesis. And you're going, whoa, wait a minute. Eukaryotic cells synthesize nucleic acid. Well, they go about it a little differently. And so that's why ciprofloxacin can attack nucleic acid synthesis in prokaryotic cells and leave your eukaryotic cells alone. Sulfonamides, which are sulfa drugs, attack folate synthesis. Now this is also specific to at least non-animal cells, because animals, we don't have the enzymes to make folate. So when the sulfa drugs attack these enzymes, it leaves our cells alone. Now, folate is used in nucleic acid synthesis, so I have a tendency to think of that as inhibiting nucleic acid synthesis. We're just attacking it at a spot that is not found in eukaryotic cells. With ciprofloxacin, or well, it's called Cipro, that's the abbreviation. With Cipro, it attacks DNA polymerase that is specific to prokaryotic cells. DNA polymerase is similar to eukaryotic cells in prokaryotic cells, but they're different enough that we can attack those separately. Because prokaryotic cells are so different, that's why we can attack these things that are specific to prokaryotes, and that's why we have so many more antibiotics than we have antifungal agents, antiprotozoal, and anti-worm and antiviral agents. So let's go ahead and move on to these that we kind of have left over. Let's start with amphotericin B. It attacks ergostol, which is a cousin of cholesterol, but it's different enough that it can bind to that and punch holes in fungal cell membranes. And, generally speaking, leave animal cells alone. I keep circling the worms because, they're animals, but, we're not worms. Quinine, which used to be effective against malaria, now most malarial strains are resistant to quinine, but we're not sure how it works, so I'm going to skip it. Now, sulfonamides, sulfa drugs, are effective against some protozoa, just like it's effective against bacteria. So I'm going to draw a line to metabolic function, and keep in mind that it inhibits folate synthesis. So what's going on here is there are protozoa that do have the enzymes for making folate, which, by the way, is a B vitamin. When you take B vitamins, you're getting folate. Then, when they can't use those enzymes because the sulfa drugs have inhibited those enzymes, they don't have the transport proteins in their membranes to bring in folate from the outside. So it kills them. Animal cells, on the other hand, because they do not have the enzymes, for folate synthesis, they do have the proteins to bring in folate from the environment, because otherwise we wouldn't be able to make our DNA and our RNA. So that's why sulfa drugs show up in both the antibiotic and the antiprotozoal categories. Now, this one here inhibits microtubule synthesis in worms. Now, that I would consider to be a metabolic function. And the plan is that you inhibit microtubule synthesis and you keep the cells from dividing as the worms are developing. Okay. Now, could that cause a problem with people taking it? That's probably part of why it's not used in humans in the United States, although why they're still using it veterinarily, I don't know. Now let's go over to our antivirals. Acyclovir and this one, which is basically Tamiflu, that's the brand name, inhibit enzymes that are specific to viral function. So acyclovir inhibits nucleic acid synthesis. The plan is, with acyclovir, is that the virus is replicating more rapidly. It's making more of its DNA, more of its RNA in the eukaryotic cell, that when we shut that down, we can kill, unquote, they're not really alive, the virus without killing the cell. That's the plan. Now with Tamiflu, it inhibits uncoating and penetration of the virus. Now these are specific to viruses, trying to keep the DNA, or actually in this case the RNA genome, from getting into the cell. On to side effects. Now the most severe side effects are going to show up where you have potential crossover of targets. So what do I mean by that? So if you're targeting something that's in a eukaryotic cell, you're going to have a higher potential of severe side effects because there's not as big a difference between eukaryotic cells and prokaryotic cells. And there's also, because we're attacking something that is inside of a eukaryotic cell, namely your cell, then there's also going to be a potential for severe side effects. So, specifically, if I am attacking nucleic acid synthesis with, say, acyclovir or with ciprofloxacin, there's a higher chance of having severe side effects. And if you go back and take a look at the table, you're going to see that reflected. Also, amphotericin B has severe side effects because ergostol isn't all that different from cholesterol. So the plan is that you're going to have lower toxicity in animal cells than in fungal cells, but they do only administer amphotericin B in the hospital so that they can monitor this. And as I mentioned before, this one is no longer used in the United States for human treatment, and it's probably because there's too much crossover between inhibiting the microtubules in the worm larva versus attacking the microtubules in your cells. Now, another reason why there could be severe side effect is systemic delivery. If you're taking it orally or if you're doing it IV, then all of the cells in your body are being affected. Whereas if you deliver it topically, you can use something like, say, amphotericin B, if it were put in a cream, and it would have lower toxicity, lower side effects, less severe side effects, than if you were taking it orally. So when you go to the store and you buy those antifungals that you're putting between your toes for the athlete's foot, it's an over-the-counter because the potential for side effects is much less. Now, the last thing, most common side effects of antibacterials, gastrointestinal distress. In other words, you get diarrhea, you get an upset stomach, and that's because most antibiotics attack all bacteria. They are broad spectrum. They attack more than one class of bacteria. So they're attacking your good gut bacteria as well as the nasty evil bacteria that you want to get rid of that's causing the infection. Yeah, one of these days we're currently working on getting antibacterials that are targeted to just the pathogen, and they'll leave our friendly bacteria alone. And then there's allergies. We'll be talking about that a bit later. But allergies are basically your immune system seeing this chemical, penicillin, or the sulfa drug, those are the two most common, as being part of a pathogen. And you get an immune response to it, even though it's no longer part of the pathogen. Well, hopefully this walkthrough of the information that was in the table will help you understand it. I find that if I rearrange things, then it helps me understand them in a different way. And the more ways I rearrange it, the better. Hopefully that also works for you.